Hello, my name is Bernard Ollier, and I'm the CEO of Vedanta Biosciences. Thank you, Nancy and CD Foundation, for the opportunity to share a short video on our work with your audience. What we're doing at Vedanta is we're specializing in pioneering a new modality of microbiome drugs that are based on defined consortia of bacteria. And what we mean by that is that the active agent in the drug are individual species of bacteria assembled as a consortium that are manufactured starting from pure clonal cell banks, obviating the need to rely on donors of fecal material to make the drug. The defined bacterial consortia are given in capsule form as a lyophilized powder, which the patient ingests. They pass through the stomach and they reach the intestine, where the capsule releases the bacteria, which then become metabolically active and they colonize in the intestine. Abundantly and durably, we've seen now across 3 phase 1 studies in about 140 healthy volunteers that the bacteria that we've selected for our consortia can colonize very abundantly and durably. And in the context of certain conditions like C. difficile, that colonization is critical to be able to provide competitive exclusion of the pathogen, in this case, Clostridium difficile. Also, the right bacteria can stimulate a range of immune responses that can be beneficial in the context of other conditions like IBD and cancer, for example. There are multiple advantages to, we think, to being able to use defined consortia instead of having to rely on fecal approaches. The first one is the ability to truly rationally select the bacteria that will be included in the drug, as opposed to just picking anything that grows or that comes out of a patient's donation. And something that we've done at Vedanta is a deliberate effort to go one step beyond this black box that is fecal transplantation, where we don't really know who does what, and actually systematically identified specific bacteria or groups of bacteria that stimulate specific immune responses. For example, we discovered some bacteria that are very useful for inducing regulatory T cells, and those are the basis of some drugs that we're developing for IBD, for allergy, and beyond. We've also discovered bacteria that are very good at inducing cytotoxic CD8 cells, and we think that those bacteria could be of use to patients that have cancer. And here below you can see some of the publications that we put out on the work that we've done. The second considerations for using defined products are, or other considerations are safety and the ability to scale the approach. And I think that an analogy to the field of blood products is useful to understand how we think the microbiome field will evolve. So in the blood products, we started with whole plasma, which over time evolved into IVIG therapies, which involve treating the ethanol with ethanol, the blood, the plasma to get rid of viruses and other pathogens. And ultimately, the biotech industry figured out a way to obviate the need for donors and use cell banks to make biologics. And thus, monoclonal antibodies sort of became a default option for new biologic drugs, and we've never really looked back. And those had advantages when it came to safety. They couldn't transfer viruses like IVIG or plasma, and that had been an issue historically with HIV and Hep C. And also in terms of scalability, once you know how to bang and ferment, you can make as much as you want. In the microbiome field, we think there's going to be a similar evolution. We started with fecal transplantation-based products where you get the whole community. And we know that there's some safety issues with those, which have become more obvious over the last year, with some deaths reported from E. coli contaminated FMT, and now with knowledge that SARS-CoV-2 is carried in the stool in a way that can, in a form that can be infectious. There's been in the field an effort to move into fractions of fecal transplants, for example, spore fractions, with some encouraging recent results. That is potentially an improvement in terms of safety because you can kill a number of the pathogens that are carried in stool, but not all of them. And we think that the next logical step in the field is to go to the fine products where we no longer have to rely on fecal material of a known and undefined composition, but actually can start from cell banks and produce as much of the drug as is necessary in a way that is truly safe and scalable. Safe because we know exactly what's in the product, which means we know what's not in there. And so the inherent safety risks around transferring pathogens are eliminated. But also, by not needing to rely on networks of donors, you can make as much of the material as you need and really bring the cost of goods down as you scale. Some examples of work that we've done using the FINE consortia are outlined here. And I'll end with this last piece of information. We have a first program for Clostridium difficile where we've put together, we're discovered the FINE consortia that can exclude C. difficile from the intestine. And this program is now supported by BARDA via a potentially multi-year contract. And also, to my knowledge, the first time that BARDA has gotten behind supporting a microbiome product. We also have a product for IBD in development that has finished phase 1 studies. The next step will be starting studies in IBD patients. That's VE302. VE303 is yet a third different consortium that we developed for cancer that is in phase 1 studies in cancer patients. And we have one more clinical study in an one more clinical program, VE416 in food allergy, that is now undergoing a physician-sponsored study at MGH on patients that have allergies to peanuts. So that's all. Thanks for the time and enjoy the conference.